This module for our flow core will be on panel design and color choices for your experiment. I am Christopher Lazarski, the director of the flow core at Children's National Hospital. We're going to explain how to distinguish between different fluorochromes and choose which are suitable for your experiment. And in this module, we're going to discuss terms including intensity, wavelength, excitation and emission, and spillover. Some topics which we will discuss include selecting fluorochromes, choosing available channels, making sure that the fluorochromes are not overlapping, and pairing antibodies with markers of interest. When selecting fluorochromes, it is important to understand the different relative intensity between different fluorochromes. Fluorochromes can be divided into bright, medium, and dim intensity markers. Some of the brightest markers include PE, brilliant violet 421, and APC, while some of the conjugate markers are not as bright. These could be classified as medium intensity. Some of these include PerCP Cy5.5, APC Cy7, and PE Cy7. These are possibly medium intensity because of the increased level of background. Markers which could be classified as dim would include FITC, Alexaflor 700, and unconjugated straight PerCP. Some fluorochromes can have a brighter alternative or one with less spillover. These are manufacturer-specific fluorochromes. One example is Brilliant Violet 421 and Pacific Blue. They both operate within the same channel on our Cytoflex instruments, PB450. However, Brilliant Violet 421 is brighter than Pacific Blue and would be preferred. Another example is APC R700 as compared with Alexaflor 700. Both of them, again, operate within the same second red channel on our instruments. However, APC R700, as we can see here on this manufacturer chart by BD, is brighter than Alexa 700 and, again, would be preferred. Intensity is defined by the difference between the brightness of the positive peak and the brightness of the negative peak. The spread of the negative peak can hurt the resolution of your signal. Besides intensity, it is important to understand which detectors are compatible with certain fluorochromes. In this example here, some fluorochromes occupy the same channel and are incompatible. FITC and Alexaflor 488 both occupy the same channel and would therefore be incompatible in an experiment on our Cytoflex instruments. Similarly, PerCP, PE Cy5, and PerCP Cy5.5, again, all emit light within the same approximate range and would be incompatible if placed within the same experiment. APC and Alexaflor 647, again, are examples in the red channel which occupy the same channel. It is important for users to understand what fluorochromes they're using and which channels are available and compatible on the instruments that we have. In addition to the brightness of the fluorochrome, the intensity also depends upon the density of receptor expression on the surface of the cell. Some markers are very highly expressed, such as CD4 and CD45. Both have more than 100,000 molecules per cell, while other surface markers on T cells are not nearly as expressed, such as CD28 or CD7. Additionally, it is important to know which markers are co-expressed on the same cell when selecting fluorochromes which occupy neighboring channels. CD3, CD4, and TCR alpha beta, for example, all are expressed on T cells, and users should be aware of the biology of their cells when selecting available channels to place these co-expressing markers to avoid certain channels with a high degree of overlap. Again, it's important to understand the relative expression level when selecting your antibodies in order to pair receptors with low expression with bright fluorochromes in order to help clearly identify those markers from the background. Another important characteristic of fluorochromes is their spillover. This is because an inherent property of fluorochromes is that they emit light across a broad range and potentially spanning multiple detectors. In this example, FITC emits light ranging from 500 all the way out to 650 nanometers. This is well beyond the detector that FITSI has and spills over into the neighboring PE detector. This is important because spillover matters for multicolor experiments. There must be some sort of correction for this intensity in the neighboring channels, because otherwise you may not be able to tell the difference between a highly expressed signal in the FITSI channel, which could be GFP from a transduced cell, spilling over into a marker in the PE detector, which is not nearly as bright or is very dim. Spillover depends upon the fluorochrome, the filter sets for the instrument, and the cytometer gains. Some fluorochromes have complex spillover, and this is often seen with fluorochrome conjugates. In this example, 16.4% of the possible PE Cy7 signal can potentially leak back into the PE channel. The excitation and emission of Cy7 relies upon energy from the PE molecule. However, not all of the PE energy is transferred during FRET, and therefore you get some leakage back into the PE channel. It is best practices for users to perform single color staining controls using the antibody in question and not a substitute. This is to account for possible lot-to-lot variation in the conjugation of the PE and Cy7 molecules. It is important for users to look up the manufacturer's specific characteristics for the fluorochromes in question, because manufacturers can often have proprietary fluorochromes. This is a sample chart from Miltenyi. Miltenyi has their proprietary VIO fluorochromes, and if using one of these VIO conjugates, it would be important to understand the difference between PE VIO 615 and PE VIO 770, for example, and the manufacturers will provide this kind of information to understand which channel would be appropriate for the fluorochrome to pair with. Please confirm that any antibodies and fluorochrome markers work with our instruments and the available channels on our Cytoflex instruments. And also, please confirm that old vials of antibody, which you may have, still function properly and have not degraded over time, as conjugates can often break down over time even at 4 degrees. For our Cytoflex instruments, we have a non-exhaustive list of the fluorochromes and their compatibility with each of the 4 possible laser channel configurations. Our Cytoflex instruments can have up to 5 channels off of the blue laser, and these are labeled FITSI, PE, ECD M Cherry, PerCP PC5, and PC7. For our red laser, the Cytoflex instruments usually have 3 available channels, which would include APC, APC Alexa Fluor 700, and APC A750. For the violet laser, our Cytoflex instruments can have either 4 or 5 available channels. Both Cookie and Oscar have PB450, KO525, Violet 610, Violet 660, while Oscar has the 5th channel, Violet 780. Finally, Cookie has a yellow laser, and it has 4 available channels on it, including ECD M Cherry, again PE, PC5, and PE Cy7. The yellow laser would be suitable for detecting red fluorescent proteins, as it activates the RFP, DS red, or M Cherry molecules much more efficiently than a blue laser. This next slide is to help demonstrate the spillover characteristics for 5 fluorochromes off of the blue laser. This is to help visualize why certain color combinations have more potential for spillover than other color combinations. What is apparent is that PE and PECF594 have slightly overlapping filter sets. This is why you have spillover from PE into the PECF594 detector. Because of this high degree of overlap between these 2 channels, users should take care when selecting a bright PE marker with a comparatively dim PECF594 when co-expressed on the same cell. BD on their website has provided a numerical translation of this spillover for each of the laser and filter combinations. If we translate this into numbers, we can see the spillover into PE from FTSE is approximately 12%, and the rest of the categories spilling over into PE are expressed across in rows. The spillover from PE into the other channels is expressed in columns, and we can see that at least 12% of the PE will spill over into the PECF 594 channel. Although these numbers are not compensation, because compensation depends upon the gains that were used when collecting, it is important reference material which BD has provided and other manufacturers can provide as well for better understanding of neighboring channels which have complicated spillover. Continuing on to the red laser, we can see how APC spills over into the AlexaFloor 700 channel, and AlexaFloor 700, nearly 15% of the total intensity spills over into the APC Cy7 channel. Moving on to the violet laser, we can see the relatively broad emission patterns for the various brilliant violet dyes and the comparatively narrow filter ranges which we have available on our instruments. What we can see is that for BV605 and BV650, that the potential for spread and spillover between these two channels is very high, and we are not necessarily even capturing the peak of intensity for BV650 because BV605 has a continual shoulder which would also be read in by that shifted filter set. Users would be advised against selecting co-expressing markers for BV605 and BV650 because the necessary compensation in order to distinguish these two would become challenging. For the yellow laser, we can again see spillover characteristics between PE, PECF594, and how nearly 20% of the PE Cy5 signal is read out by PE Cy7. Because of the limited number of channels we have available on our Cytoflex instruments, users should make several considerations when selecting fluorochrome marker combinations for their panel design. Users should consider the intensity of the fluorochrome and try to match high intensity markers with either priority antigens or antigens with low expression level. Also, manufacturer availability of specific antibody marker pairs can dictate which channels are used in an experiment. Users should also consider the biology of their experiment when selecting which fluorochromes will be used for co-expressing markers compared with alternating fluorochromes between different cell subsets. This can help minimize spillover artifacts seen between channels. Finally, some antibody clones which manufacturers supply may not be as high avidity as other antibody clones or might suffer from artifacts during fixation. And while these recommendations seem like a long list and may be overwhelming for new users to flow cytometry, the core does have recommendations for panel design which can help alleviate many of these problems. Start by identifying which instrument you are going to use and what channels are available on that machine. We would also recommend identifying the size of the panel which you are going to design for your experiment and recommend a less complicated panel which answers the questions that the experiment needs rather than a more complicated one to begin with. I know it is tempting for users to try to answer as many questions as possible within one experiment and add multiple markers. However, increased complexity will always require a more complicated compensation and a potentially more challenging interpretation of the data. This leads into ranking your markers by the priority for your experiment. The top rank markers are often activation markers such as interferon gamma or PD1. This could also be a rare marker which is of interest for your experiment. Second tier markers can include phenotyping markers such as CD3 or CD19 in order to characterize the cell types that are present in your sample, while the bottom tier can often be viability, which is available in multiple channels, or additional markers which may be collected for informational purposes but are not the primary end goal of the experiment. Next, I would recommend that users outline a gating strategy for their experiment in order to identify which critical comparisons are going to be made and which markers are going to be plotted against each other on the same graph. In this case, we have a sizing gate to begin our gating strategy, which is forward scatter by side scatter. This leads into a singlet gate, which is then followed by a viability gate. Viable cells are then phenotyped according to their surface markers, including CD3 for T cells or CD19 for B cells. Negative for CD3 and CD19 may then include CD56 for NK cells. Those T cells can then be subdivided into CD4 and CD8, and then off of the CD4s, we can then find regulatory T cells, which then express CD127, CD25, or the activation status of the T cells by cytokine production, such as TNF, interferon gamma, or granzyme B. This gating strategy highlights then important comparisons, such as CD4 at CD8, CD25 versus CD127. While CD4 and CD8 would not be co-expressed, CD127 and CD25 would be co-expressed on the same CD4 T cell, and for this receptor pairing, it would not be advisable to choose neighboring fluorochromes such as BV605 and BV650 for CD127 and CD25. This is the next recommendation to avoid placing co-expressing markers on channels with high spillover. In this example, if we are looking at FOXP3 positive T cells, which are CD3 positive, CD4 positive, TCR alpha beta positive, and FOXP3, we would not advise selecting PerCP Cy5.5, APC Alexis 700, and PE Cy7 as your 4 fluorochromes. Instead, we would suggest an alternative for fluorochromes to include FITC, PE, BV421, and APC. In this way, PerCP Cy5.5 spills over into PE Cy7, and for a co-expression panel, this is not recommended, much as APC spills over into Alexis 700. Also, PerCP and PE Cy7 are not as bright as PE and BV421. In this way, we are both selective when choosing markers with substantial spillover, and we are also selecting markers with a high relative intensity for our small panel. Also, some cross-laser combinations are not ideal for co-expressing markers because they have similar emission wavelengths and may produce a fishtail or trumpet effect, which is a spread of the data when creating your final XY plot. APC, BV650, and PerCP Cy5.5 all have similar emission wavelengths and can show this fishtail effect. Our goal for our sample panel is to phenotype lymphocytes and identify common activation markers. This sample panel would move back and forth between T cells, B cells, NKs, and monocytes. We are also pairing dim markers as well as important interferon gamma marker with a bright fluorochrome while minimizing neighboring channel overlap. We have selected CD25 to go onto BV421 and interferon gamma on PE, with APC taken up by TNF alpha. We also have alternated our markers in the violet channels to move from T cells with CD3 to B cells with CD19 back to T cells with CD4. For the blue laser, we avoid overlap between PE and PE dazzle with CD56 in the PE dazzle channel. We also have monocytes in the PerCP going back to T cells with PE Cy7. Finally, for the red laser, we place an informational marker, gamma delta, in the Alexa 700, which will have no spillover with our Treg marker CD127. Finally, we would suggest asking for help if you are unsure with how to design your panel, as we will provide assistance and help you select markers and choose available channels on our instruments. In summary, there are many possible fluorochrome conjugates, each with its own spectral characteristic. Users should invest time to design a panel which helps satisfy their experimental needs and look up manufacturer specifications for their fluorochromes, as well as identify which instrument they're going to use so that They can plan out a gating strategy to see which fluorochrome pairs will be needed and which may directly match against each other in the final data. Organize your antigens by priority and pair high priority or dim antigens with bright fluorochromes, and try alternating channels for surface expression to minimize spillover between channels for markers co-expressed on the same cell.